Hello. This lecture video is part of the biological molecule section of Module 2. So let's do a review to begin this video. You should have already watched the video or attended class to learn about dehydration and hydrolysis reactions. So I want you to remember that dehydration reactions, a covalent bond is formed between two molecules and a water is released. Hydrolysis, water is added to the reaction to break covalent bonds. So dehydration goes from small molecules to large, whereas hydrolysis goes from large molecules to small. Now, the second part of this PowerPoint was on carbohydrates, and I made a different lecture video for that portion. So what I want you to remember from the previous lecture video, hold on one. Okay, I just returned to the video. I guess dog barking is a part of filming videos at home. So from the carbohydrates lecture, I want you to remember the three monosaccharides. Remember, those are also monomers or simple sugars. So we have glucose, fructose, and galactose. I asked you to remember three disaccharides and the components of those. So the three disaccharides are sucrose, lactose, and maltose. Finally, you need to remember the three polysaccharides I taught you. So that means many units put together. And we have glycogen, starch, and cellulose. And you need to know the functions for each of those polysaccharides. So now let's move on into proteins. This is the second group of biological molecules. Remember, there are four groups, being carbohydrates, proteins, lipids, and nucleic acids. Now, for proteins, you need to be able to identify the monomer or molecule that forms proteins. You also need to be able to describe some functions of proteins. So let's get started. Now, you've seen this molecule before when I was talking about dehydration and hydrolysis. So the monomer of a protein, let me move this box here. The monomer of a protein is an amino acid. So here is an amino acid. Remember that a monomer is not a single atom, it is a molecule. So in this amino acid, we have our nitrogen. And remember, a line means a covalent bond. So nitrogen to carbon to another carbon. So what I'm showing you there are lots of different elements or atoms involved in making the amino acid. So in this box on your lower left-hand corner, I am showing you how two amino acids are joined. Now you should know right away that this is dehydration. Remember, dehydration forms covalent bonds. So here we have one amino acid. Here is the other amino acid. One loses an H, one loses an OH to form water. Now you have a covalent bond in the middle. Now you may be wondering why it says peptide bond. Well, just as with the terms monomer and polymer, covalent bond is a generic term. And when we're talking about the four types of biological molecules, we give those covalent bonds different names. So for a protein, the covalent bond is called a peptide bond. So the polymer of a protein, many amino acids together, is a polypeptide. Remember, poly means many. Now that's in contrast to carbohydrates that have polysaccharide. So what I'm showing you on this screen, there are 20 amino acids that are found in nature. So those 20 amino acids in all different combinations, all different chains, number of amino acids in a chain, form different proteins. Now, a protein could have like 100 amino acids in its chain or like 1,000 amino acids, right? It varies depending on the protein. Now, you do not have to know all of these different amino acids, but I want to point out a couple things here. So actually, I'm going to go back to the previous screen. Remember, in the upper right-hand corner, this is our amino acid, okay? Now, notice that there's an R coming off of that carbon. Well, there is no element with the symbol R. That R means a side group. So this structure here, the N with the two H's, the carbon in the middle, the carbon on the end, that is what we call the backbone of the amino acid. That is the same for every amino acid. You will always see that backbone, but the R group is different for each amino acid. So I'm looking at the top row here under non-polar amino acids. You'll see our first one, glycine, has a hydrogen for its R group. Now, all of the R groups are highlighted here. Now, alanine has a carbon bonded to 3 hydrogens. That's how I know it's alanine. Now, like I said, you don't have to know all these amino acids, but you do need to know that it is the side group that gives the amino acid its properties. So all the amino acids in this first row are non-polar, meaning that they have a lot of carbons bonded to hydrogens in that side group. The next row down, all of these side groups are polar, meaning that they are attracted to water. They have a region that's positive and a region that's negative, right? 2 different poles. Finally, we have what they call electrically charged. And all that means, if you notice on this first one, aspartic acid, you'll see the oxygen has a negative charge on it. Now, you should know that a negative charge means it has an extra electron. That's going way back to our chemistry that you learned prior to this lecture. So there are 20 different amino acids that make up proteins. Oops. There we go. Sorry about that. My computer is responding slowly. Okay, so proteins have many functions. So I selected 4 that I think are important functions to know. So the first function is to act as an enzyme. Now, you'll notice here that the function says to catalyze biochemical reactions. So let me write down what that means here. I'm going to simplify that a little bit. Okay, so first of all, I'm going to write enzyme here. When I say catalyze, that means that it gets the reaction started. To catalyze something is to allow the process to begin. And biochemical reactions are those reactions that occur within your body. So enzymes will say start, and they actually speed up chemical reactions. And then just to remind you, biochemical reactions occur in your body. Enzymes are really, really important for life. Without enzymes, the chemical reactions in our body would occur so slow that we wouldn't be able to function. Now, the next function is structural. So in your body, you have all of these protein fibers that exist outside of your cells. We call that the extracellular environment. So these protein fibers help anchor cells in place and they also give them structure. Remember that animal cells do not have a cell wall, so they need other structures to support them. So we're going to say structural, and I'm going to say protein fibers. How about provide strength and structure to animal cells? They are found in the extracellular environment. Now, if you don't know what extracellular means, that's fine, but you should define it in your notes. So extracellular environment means outside of the cell. And that's actually a good practice as you're studying any topic. If you don't know what a term means, don't just write the term down and move on. You need to define it so that you know what the information actually means. Okay, next we are looking at transport. Now, a perfect example of transport is with hemoglobin. So your red blood cells have hemoglobin inside, and this hemoglobin is used to carry oxygen throughout your body. So when we say transport, what I mean is carrying molecules from one place to another. So I'm going to write down this example. So hemoglobin in your red blood cells carries oxygen throughout the body. Now, finally, a fourth function of proteins is motor. And the one part I want you to remember, motor can refer to different things, but the example I'm going to use is muscle contraction. Proteins are necessary for your muscles to contract. So when I say motor, I mean that those proteins are moving your muscles. So for motor, proteins allow your muscles to contract. Now, just another note on studying, you do not have to write these down word for word. It's actually better if you review my video and write these things down in your own words. That way you can make sure that you understand the information. Now, proteins have several different, what should we call them? Several different types of shapes, right? We call them primary, secondary, tertiary, quaternary. I don't need you to know those, but what I do need you to know is that when a protein is first synthesized or produced, it's basically just a string of amino acids. Think of it like beads on a string. Now, the sequence of these amino acids is different for all these different types of proteins. Also, like I told you before, the number of amino acids is different. Now, what is important about this sequence of amino acids is that the side groups determine how the protein will fold. So proteins are not functional until they fold into a certain shape. I'm going to write some of this down for you. Now look at the protein on the right side of your screen here. That has like spirals, we call those helices, and all kinds of bends. Well, that is a folded protein. And the way that it folds is dependent on that original amino acid sequence. Now, also, when proteins fold, that kind of determines their function, right? An enzyme fits specifically with a certain molecule. It's like a lock and key. So if that protein, if the structure is disrupted, it's not going to work anymore. So let me summarize some of that for you. So proteins, actually, I'm going to say protein shape is determined by the types of amino acids in the primary sequence. Remember, proteins have primary, secondary, tertiary, quaternary structures. Secondary, tertiary, quaternary are all folded. So what I'm going to write here is that proteins must fold to be functional. They fit like a lock and key with the molecules they are, I'll say, affecting. Right, so an enzyme is going to bond with a certain molecule, and it's like a lock and key fit. So if that shape is disrupted, the protein no longer works. Whoops. You guys will have to excuse my grammar when I type out these notes. When I try to write, I usually correct my grammatical errors after I get done writing. So you guys get to see all my grammatical errors. I love putting commas. I put commas everywhere. Okay, so what I told you here in this slide is that the amino acid sequence in a protein determines its unique shape and function. There we go. Okay, so what I'm going to do with this slide is give you an example of what a genetic mutation can do. So I'm going to write down a little bit of information here about how your DNA or your genes are related to proteins. So genes are segments of your DNA that code for amino acid sequences. So remember, I think I told you before, about 95% of your DNA is not, does not make up genes. So your genes are only about 5% of your DNA, which is really crazy when you think about it. So much of your DNA controls when your genes are expressed, and a lot of it does things we don't even know about yet. So looking at the function of your DNA is an area of really exciting research right now. But your genes are what code for or determine the proteins in your body. And the proteins in your body are responsible for most of your characteristics. Eye color, hair color, skin color, height. All these things are determined by proteins. So what happens if there is a mutation in your DNA that is within a gene? Well, it can disrupt the shape and function of the protein. It doesn't always, but it can. So if there is a mutation in your DNA within a gene, it can, I'm going to put change the amino acid sequence of the protein, disrupting its shape and function. So let's look at an example of that. Here we have hemoglobin. Okay, and what you're looking at here is a disease you've probably heard about, sickle cell anemia. So here on the left is the correct amino acid sequence. Now notice it says 4, 5, 6, 7. I'm only showing you 4 amino acids in that sequence. It's actually much longer, right? So let's say there is a mutation in your DNA within a gene that codes for hemoglobin. And so number 6, you have the amino acid glutamate or glutamic acid, GLU. And you do not have to know the names of any of these amino acids. But let's say we have GLU for position 6. We get a mutation in our DNA, and now all of a sudden, that number 6 amino acid is changed. Now it's valine or VAL. What that does, so remember, hemoglobin is in your red blood cells. So with one mutation, one change in the amino acid sequence, that hemoglobin takes on a different shape. And these hemoglobin molecules kind of stick together and they crystallize, just meaning change shape. But what that does is it changes the shape of the red blood cell. So right here it says sickled red blood cells. That's because those hemoglobin proteins no longer have the correct shape. So what happens when you have sickled red blood cells? Well, 2 things. They do not carry as much oxygen, so your body can become depleted of oxygen, like your organs, things like that. Also, these red blood cells can stick together and clog up your blood vessels. So when someone with sickle cell anemia is having like a flare up, that's what's happening. And this occurs because of one change in the amino acid sequence. So this shows you how important amino acid sequence is to the functioning of the protein. So that is all I have for the protein section. I want you to remember that the monomer of a protein is an amino acid. Many amino acids together forms a polypeptide, which is just a polymer of a protein. Okay, I'd like you to remember the 4 functions we talked about. You should also know that your genes are what code for the amino acid sequence of a protein. And it is the amino acid sequence of the protein that gives it its shape and function. So my next lecture video will be on nucleic acids.